Welcome back. In today's lesson, we're going to be talking about roots and doing the same sort of parallel comparison between monocots and dicots. So let's dig in. Roots are the underground vegetative portion of a plant. I think that's probably pretty obvious. Some of the functions that roots play are anchorage, right? That you got big trees that are holding themselves upright. The roots also deliver water and nutrients, right? That's important. They also can be used to store things like sugars and nutrients from different times of the year. Um, and sometimes there's adaptations to the roots that help to stabilize plants. So, just a couple of slides looking at some erosion. This is a mango tree and kind of shows the typical root structure of a tree. Um, Here we have sort of a what we call a tap root. This is a cotton plant. It's not a very good picture, but it's free. um, this is a ficus tree. Really interesting adaptation here. You can see where this gentleman's standing. The roots are very tall and vertical, and they're on top of the ground. That's an adaptation for trees to exist in ground that's very soft or friable and doesn't offer a good anchorage. So in addition to entering the ground, the roots also build structures above to further support the plant. Then we have roots like a mangrove, where they have to both prop the plant up so that it's above the water line, and they have to be able to filter the water and funnel oxygen down. And mangrove roots are really fascinating, what they're capable of doing. And then there's sort of unusual things like stilt roots. These are sort of, as I was saying, where plants can make adaptations to prop themselves up. Typically, again, in poor or loose, friable soil that won't anchor as well. So the plant sort of spreads itself out. Why? Because the plant's got to get as high as possible into the canopy to outcompete its neighbors for light. Then there's classic monocots. This is a royal palm. And in the back right-hand side, you can see the roots as they sort of come out of the trunk and spread into the ground. Adventitious roots, and then ultimately the main roots. These kind of root systems are a lot like a bundle of straws. I said that when we were talking about stems, and that it applies right through the stem and right into the ground in the sense that you get the this sort of flexible bundle of fibers that allows the trees to adjust to those really high winds. Now, if you look at the root itself and go to the very tip, let's start there and look at how that its form and function. Um, right off the bat, this purple stuff that you see, let me get my pen. This purple hue that you see here, this is actually a dried mucilage cap. So what happens is, as the root is growing through the soil, the plant wants to protect the delicate growing tip or meristem. And so it has a couple of strategies. One is this, it exudes a mucilage. And I have a picture of a snail down here because snails are crawling in a mucilage that's very similar, so that they can slide over something like, in this case, asphalt. Um, in fact, they swim in their own slime. That's what they do, right? There's like hundreds of thousands of little legs paddling away in the slime that they produce. That's how they move. Anyway, the mucilage for the root protects this emerging tip and allows it to push through the soil and against abrasive structures as it navigates what's in ground, which is, obviously a lot of broken down minerals, organic matter, and things like that. And as it's bumping its way through the soil looking for water and nutrients, it needs to be protected. And so the root cap offers that. Behind the root cap is, or behind the mucilage is the root cap. Sorry, I got that flip. And the root cap, essentially, if you see in this picture here, there's these cells that are just sloughing off. They're loose all over the place around the tip of the root. And these are sacrificial cells. You'll notice there's not a lot of structure to them. they're pretty wimpy, and their job is just to be basically like a crash barrier. As the roots pushing through the dirt, these cells take the damage and just slough off, and they're quickly replaced. So right behind here, we have the apical meristem, and it's rapidly pushing out these root cap cells to protect itself, and it's leaving behind the important cells that form the permanent root structure. In a lot of ways, it's the same thing actually happens in your mouth. If you take a Q-tip and you slide it on the inside of your cheek, you get cheek cells, which look very similar. They're sort of wimpy, baggy, clear cells, not much structure. And that's because as you are talking, like myself, your jaws are moving up and down and they're scraping the inside of your cheeks. And your body doesn't want to have too much damage there. So it has this sort of sacrificial layer of cheek cells, just like a root cap cell, that will constantly slough off to protect the important dermal cells underneath. So, summarize here, we've got a mucilage root cap, and we have a mucilage, and we have a root cap full of cells that are sacrificial and that slough. Moving on up the root, we look at root hairs. So there's lots of little hairs coming off this root. Most roots have root hairs, pretty much all of them. They're just different densities and lengths. This is a close-up of a root hair. And what I want you to observe here is just how sort of thin the cell wall is. It's like a saran wrap bag. It's just this thin, precarious little hair sticking out into the soil. And it's that property of the root hair that gives it its most important role, which is to take in water and nutrients, right? And because it has such a thin cell wall, it's very effective at that. It brings in water and nutrients very quickly. And it can also stretch out very quickly into the soil to areas where there's interesting nutrients. So right here it says roughly 60% of water is absorbed through root hairs. That is true, and it's important to understand because when you're growing plants, we have to make sure that we take care of our root hairs. If you let the soil get too dry, the root hairs die. So you would lose all these root hairs, and you would just have the root, which means you've lost 60% of your ability to take up water and nutrients. And it takes about 3 weeks. Oh, can I write with my mouse? Kind of. It takes about 3 weeks to regrow root hairs properly. So that means you have 3 weeks of 60% less water and nutrient uptake. So damaging your roots is not a good idea and leads to all kinds of problems with diseases and nutrient deficiencies. Here's a close-up of some microgreens germinating. You can see lots and lots of root hairs. It's obviously a very humid environment there, which is convenient for us because we can see them. A lot of times these root hairs wouldn't be out in the air like this. They would be in the soil and just infiltrating all the organic matter, trying to find nutrients and water. So looking at the structure of roots, we have two types. We've got a tap root and we have a fibrous root. This is your quintessential tap root. So it goes straight down like a spike into the ground. You notice it tapers to a point, and then it has lateral branching. And the fibrous root is just like a mess of randomly oriented root fibers going in all directions. So typically we get fibrous roots with monocots, and we get tap roots with dicots. That's generally how it goes. With a tap root, we have a seminal or central root, which continues straight on down into the ground. And then we have orders or branching roots coming off the side, first, second, third order branching. So it's fairly structured. Whereas in the fibrous root, you'll notice that there is no dominant central root, right? So they start and then they quickly split off. It's like sparks or a firework, right? They're going in all directions, constantly branching and growing. So that's a fibrous root. Are there different advantages to the different ones? Yeah, yes and no. The taproot kind of gives you that deep, firm anchor that the plant can hold on to. It can also get down fast to where there's water, so in drier conditions, that sometimes is advantageous. Um, but that's not necessarily essential because grasses are often very drought tolerant plants and they have a fibrous root. So, um, looking at root structure, together my little icon here, uh, looking at root hairs, right? So the hairs are coming off, epidermal tissue in the exterior, the stele, that's an introduction to a term. That's the central column that includes the vascular bundles. The cortex, right? That's this sort of area just outside the stele. The apical meristem, this is where the heart of it all is, right? This is where all the division's happening. This area is busy dividing and it's laying down root behind it, just as if it were like a machine laying down a new road. It's slowly advancing in this direction and it's leaving behind all that structure of the root. So this is a very busy, important brain part of the root. And then there's the root cap, which is protecting, like a helmet, protecting this important part of the meristem. So lucky for you, I've got a better organized bunch of arrows. Let's take a closer look at the vascular cylinder or the stele. I've circled it with a red ring here. It contains both xylem and phloem. So just like in the stems, the general rule is if you see big holes, these are xylem, right? So that's your X. I put X's here. So these are xylem. And they're moving your water and nutrients around, up from the bottom of the plant up to the top. And then you have your more dense phloem areas, right? So you got phloem here, phloem here. Xylem's in the middle, phloem's in the out. And then between them, let me switch colors. Between them, we have the meristem, or in this case, we'll call it a cambium, right? So it's dividing and it's laying down xylem on the inside, phloem on the outside. Now that is all surrounded by something called a pericycle. Okay, pericycle. That's the next layer outside. The pericycle is where you get lateral roots. So see how this is a root that is forming and growing out the side into the soil. These cells have originated from the pericycle. Let me get a different color. They've originated from the pericycle. So this layer just outside here, and it's growing out. And this sort of outer boundary follows along and continues to enclose it along with the vascular cylinder. Then there's the cortex. The cortex is this fleshy tissue out here where the plant or the root is storing starches. It provides structure, protection for the cambium or for the vascular stele. And then there's also epidermis. So epidermal cells are where your root hairs come from. That's an important one to remember. So the outermost layer here, this outer layer, these epidermal cells will spontaneously evolve into a root hair. Some of them, not all. That depends on the plant's genetics. And so that's where the root hairs come from. So we've got a dicot root. In this case, we have the epidermis, right? That's the outermost layer. We have the cortex, which is this region between the epidermis and the stele, right? This region here. And then if we zoom in on the central stele, we have the xylem and we have the phloem and we have this cambium that's between them. In this case, it's sort of like little happy face arches between them. It's a fairly young root, so they haven't, the vascular cambium hasn't grown together into a ring in a more mature, as it would in a more mature, larger root. So it's okay to have them in chunks. When you're looking at your slides, you'll have to try to find the region. So you see there's the phloem, and there's the xylem. You know that the cambium is going to be between them, or the vascular meristem. Then there's the pericycle. Hard to see this. I'm going to go to the next slide to show you that. So the pericycle is on the inside of this band that surrounds the vascular bundle, and the endodermis is on the outside. So we'll talk a little more about the endodermis soon. So here's a review slide showing all the different structures of the dicot root with the epidermis, the cortex, which is storing starches and nutrients, the vascular bundle, the stele, where we have the phloem and xylem, the pericycle, and the endodermis. Here's another slide. These are just to help you remember if you're doing your studying. Picture's worth 1,000 words. You don't have to listen to me all day long. Let's look at a monocot root. Very different. There's this sort of ring structure, which is interesting because in the stem, if you recall, the vascular bundles were randomized all over the place. Remember, they were like bundles of them, all oriented with phloem to the outside, xylem to the interior, but these bundles were all over the place, right? And we talked about straws and bundling them. When you look at the root, at some point near where the plant transitions to soil, all these bundles organize themselves and rearrange into a ring, a solid ring. So it's a fascinating, interesting difference. Other than that, there really is the terms are the same, the locations are the same. So you have epidermis, you've got your cortex, you've got your endodermis, you've got your pericycle, you have xylem, and you have phloem, and a cambium. Let's look at the next slide. It'll kind of help. So here's the monocot at the bottom. The cortex is exterior to the stele, as I mentioned. Then there's this endodermal layer. Coming up next, we'll talk about that. Then there's the pericycle, which is where your lateral roots will emerge and branch from, because monocots branch their roots too. Then you've got your phloem and your xylem. And the pith, which is the center, serves no purpose in terms of conducting water or nutrients. It will somewhat conduct passively, but it's more where the plants deposit metabolic waste or store nutrients for later use. So that's a big difference between a dicot and a monocot root, is that the existence of a pith. So hopefully those differences are clear to help you navigate between monocots and dicots, right? I want to talk about how water flows into a root. The first method is called symplastic. This is where water enters through cell membranes. So this is the S for symplastic. So the water enters a root hair, passes through the cell wall, passes through the membrane, enters the cytoplasm, and then once it's in the cytoplasm, it's connected via little pores through all the other cells and eventually makes its way into the stele where the water and nutrients can then transport up and down the plant. The nice thing about the symplastic is that it offers filtration, because the water has to pass through a membrane before it can enter the plant. And the membrane, which is like a lipid layer or an oily layer, will filter out a lot of things. And the only way stuff can get through that layer is if the plant allows it, right? If we look at another method for plants entering, for nutrients and water entering plants, it's apoplastic. And with the apoplastic, with the A over here, what happens is the cell walls, every cell in a root is surrounded by a cellulose wall, right? And think of a cellulose wall as like kitchen towel, paper towel. And so if you put it down on a piece of water, the water wicks into that paper towel. And so if the cell walls are all made like that and you have water out here, the water will wick through the cell wall and just travel along as if it were in like paper towel and just wick between the cells all the way to the middle of the root. Now, that's great because it might get some filtering because of the fibers, but at the end of the day, what's going to happen here is potentially anything could get in the root, and the plants would have no way of controlling it. So to prevent that from happening, to prevent apoplastic travel beyond the cortex into the stele, what the plant does is it forms what's called an endodermis. That's the term I've been avoiding up until now. Endodermis. Okay? And that endodermis has something called Casparian bands, which basically plug up the cell walls. Now, if you look carefully, let me clean up this a little bit. If you look carefully, I'll circle, you'll see these thickened walls that are side by side in this ring that goes around the stele. You can't see this one here because I got a big X on it, but what that is it's a layer of suberin. It's sort of like lignin, it's but a little bit rubbery. And by sealing off the walls, each cell has a sealed ring that connects to the next cell. And that makes that sort of cellulose paper towel like cell wall suddenly waterproof. And so along comes your water trying to take shortcuts through the cell wall instead of going through the membrane. And it's wicking its way through and it gets to the Casparian band. And then all of a sudden, all bets are off. It can't get in the plant. The only way it can get in the plant is to divert into the cytoplasm through the membrane, which means the plant still has control and can filter it. So it's important to understand that water getting to the central stele gets filtered, but water in the cortex, not necessarily. And that's how often we get fungal infections, is because that cortex is exposed and not filtered. The water is not filtered until it gets closer to the center. Um, Talk a little bit about root adaptations, nodules, and fungal associations. Um, with the plants, they often form symbiotic relationships with other organisms. And the classic example is, soybean, for example, that's grown commercially all over Ontario. The soil has these sort of bacteria they are capable of taking atmospheric nitrogen, which is in the air all the time and actually percolates into the soil. So it's in the soil. That helps make sense of this. So atmospheric nitrogen is diffusing into the soil, and bacteria can take that nitrogen and convert it into ammonium, which plants can absorb and use to make protein and chlorophyll and things like that. So these nitrogen fixing bacteria, as we call them, are really, really useful because it improves the nitrogen content in our soil so that we don't have to use as much fertilizer. And these little bumps you see on the roots are a further adaptation where what happens is the plant senses the bacteria because it enters its root and it produces a structure, a nodule structure that protects the bacteria. And on top of that, they produce a chemical known as leg hemoglobin, which helps reduce oxygen in the center. So in the center of this sort of ball, you have low oxygen, and that helps to improve the conversion from nitrogen gas to ammonia. Okay, so we have nitrogen from the atmosphere going into the nodule, and with the help of the plant, which is providing protons and electrons from photosynthesis, it is converting nitrogen to ammonium, which the plants can then absorb. Now you might ask, why are the bacteria doing this? What on earth? Well, essentially the plant is mining the bacteria for its own benefit, and it's feeding the benefits sugar in exchange. We call that mutualism. So, or symbiotic relationship, right? So the bacteria give their services and give their excess ammonium to the plant, ammonia to the plant, and the plant in return exchanges sugar to the bacteria or carbohydrates to basically so they have a nice place to live. This chemical, leg hemoglobin, is related to the hemoglobin you find in your blood, and that's how it's used to keep oxygen levels low. And that's why often you see there's a different color in the nodule, because the hemoglobin changes its red tinge depending on whether it's bound oxygen or not. And in this case, it would be a layer surrounding the center that has bound oxygen, keeping oxygen low in the middle. Let's talk about mycorrhizal fungi. These are fungi that infect plant roots and form an intricate connection with the plant so that they can share resources. And it's estimated that 90% of plants out in the wild have some sort of fungal association. I know there's a lot of interest in root sort of biosystems, but we really don't know much about this stuff. There's a lot to be learned. But mycorrhizae are some fungi that infect roots. They they increase water and nutrient uptake, specifically phosphorus. That's the key one to remember. So a mycorrhizal association enhances water and nutrient uptake, particularly phosphorus. And just like with the rhizobium bacteria and the nodules, it's a mutualism relationship. So the plant feeds the mycorrhizae carbohydrates in exchange for the water and nutrients that the mycorrhizae brings back to the plant. Probably like the best scenario to think of is you say you have a tree in the forest and its root line is about the size of the drip line of the tree. So the width of the branches dictates roughly the the area underneath the ground that the roots occupy. If a tree were to go enter into a mutualistic relationship with the mycorrhizae, it would almost quadruple the soil volume that that tree has access to for both water and nutrients. So it's absolutely essential. Um, a lot of the times people go up north, go camping, and they see a beautiful little evergreen, and they want to bring it home and plant it. And they bring it home and they plant it, and it doesn't do very well. And it's not saying you can't grow those plants, but a lot of the times the trouble is you're not bringing along the established mycorrhizal relationship that was happening up in the north, and so the plant doesn't thrive. Um, this is what roots look like once they're infected. They kind of look like fat fingers. They look like my hand. Uh, if you're ever digging up a plant and you see that the roots are sort of thickened like that, then you know that they've had a mycorrhizal relationship. An interesting plant, the ghost plant used to be called Indian pipe, uh, Monotropa uniflora. This plant is technically what we call a saprophyte in the sense that it feeds off dead and dying organic matter, but that's not enough alone. It typically has a mycorrhizal association, right? So it lives near a tree. Oftentimes, that tree will have a mycorrhizal association already. And then the mycorrhizae actually bridge between the roots of the different plants. And this way, the ghost plant can actually access water and nutrients from the mycorrhiza and even take some of the nutrients, the sugars that the mycorrhiza has been funneling from that tree, and will use it for its own growth. And so these things have no chlorophyll. They survive just through these relationships. It's pretty cool how everything's connected in a soil environment. When it's no surprise when we take something out of that context, it doesn't always thrive. Here's a close-up of the plant's root system. Anyways, that brings me to the end. That was a shorter class, 30 minutes. But there's lots there, lots for you to review, and you'll have lots to talk about in class with Linda. Well, okay, take care.